Imagine you are looking at a map of Europe, but not a political map, a genetic one. Every dot is a person, and their position is determined not by where they live, but by who they are, genetically. Swedes cluster with Norwegians. Danes sit nearby. The British, the French, the Germans, they all overlap in a dense, messy cloud across Central and Northern Europe. And then there is Finland. The Finnish dots drift. They pull away from the Scandinavian cluster, sliding northeast on the plot, towards something that is not quite European, not quite Asian, and not quite anything else on the map. They sit alone. And the first time you see this, really see it, your instinct is to assume something went wrong with the data. Nothing went wrong. Finland shares a border with Sweden. The two countries have been politically intertwined for 7 centuries. Finnish and Swedish citizens cross the Gulf of Bothnia like it is a commute. And yet, when you compare their genomes, the Finns look less like their Scandinavian neighbors than almost any other European population looks like its own. That gap is the story. Not the cultural gap, not the linguistic gap, though those matter too. The genetic gap. And explaining it requires pulling on threads that reach back thousands of years, across thousands of kilometers, into a part of the world that most European history textbooks barely mention. Here is the thing that makes this strange. Most Europeans, genetically speaking, are assembled from the same 3 ingredients. Paleolithic hunter-gatherers who settled the continent during the Ice Age. Neolithic farmers who expanded out of Anatolia, modern-day Turkey, beginning around 8,000 years ago. And steppe pastoralists from the Pontic region, north of the Black Sea, who arrived roughly 4,500 years ago, bringing horses, wheel technology, and almost certainly the ancestor of most European languages. This 3-part model works remarkably well. It explains the genetic structure of Spaniards and Scots, Greeks and Germans. You can take a Swede and model their ancestry as a blend of those 3 sources, and the math checks out. Try it with a Finn, and the math breaks. Something is left over. A residual signal that the 3-source model cannot account for. For a long time, researchers described it vaguely as increased allele sharing with East Asian populations, which is a polite way of saying there is something in the Finnish genome that does not come from any of the usual European sources, and we are not sure where it came from. Actually, let me rephrase that. We are not sure when it came from, because by the early 2000s, the where was already narrowing. Linguists had known for over a century that Finnish does not belong to the Indo-European language family. It is Uralic, part of the Finno-Ugric branch, related to Estonian and, more distantly, Hungarian. The Uralic homeland, as best as linguists can reconstruct it, lies somewhere around the Ural Mountains and western Siberia. The language itself carried a Siberian fingerprint. The genetics suggested the same. But the old consensus assumed this was ancient, maybe Mesolithic, a faint trace from the earliest settlement of the region after the last glaciation. That assumption held for decades, and it was wrong. Not slightly wrong, fundamentally wrong about the timing, the scale, and the mechanism. Here is why the error persisted. 3 separate blind spots converged. First, the technology available through the 1990s and early 2000s could only examine tiny fragments of the genome. Y-chromosome studies looked at paternal lineages. Mitochondrial studies looked at maternal lineages. Neither gave you the whole picture. It was like trying to reconstruct a painting from 2 random brushstrokes. You could see color and direction, but not the scene. Second, the dominant model for European genetic history was built on populations from Central and Western Europe, where the 3-source model fit elegantly. Northern and Eastern peripheries were treated as complications to be dealt with later. Finland, in particular, was filed under known outlier, cause unclear. Researchers acknowledged the anomaly, but did not prioritize resolving it, because the framework that worked for 80% of Europe felt like it should eventually accommodate the remaining 20. Third, and this is the one that I think mattered most, there was no ancient DNA from Finland. You cannot date a migration without a timeline, and you cannot build a timeline without ancient genomes to anchor it. Modern Finnish DNA told you the Siberian component was there, but it could not tell you when it arrived. Without that anchor, the assumption defaulted to very old, probably Mesolithic, because that was the simplest explanation. Simple explanations are comfortable. They are also sometimes wrong. The problem was that earlier genetic studies relied on small data sets, a few hundred individuals, a handful of genetic markers. Y-chromosomal studies noticed that Finnish men carry haplogroup N1C at extremely high frequencies, roughly 60%. This haplogroup is overwhelmingly Siberian. The Yakuts carry it. The Buryats carry it. In Sweden and Norway, it is virtually absent, replaced by R1A and R1B, the classic Indo-European paternal lineages. But here is where it gets strange. And this is the part that genuinely surprised me when I first dug into the literature. The mitochondrial DNA, the maternal lineage, tells a completely different story. Finnish mitochondrial haplogroups are overwhelmingly European. Haplogroup U, haplogroup H, haplogroup V, the same lineages you find across Scandinavia and Western Europe. Think about what that means for a moment. The fathers came from the East. The mothers were already here. That asymmetry is a signature. It tells you something specific about how the population formed. Not a mass migration where entire families moved together. Something more targeted. A wave of males, likely carrying language, trade networks, and possibly specific technologies, integrating into existing European communities. And the genetic evidence sat there for years, split across different studies, different labs, different papers, without anyone pulling the two halves together into a single coherent picture. The reason nobody pulled it together is partly about tools, partly about assumptions. Y-chromosome studies and mitochondrial studies were published by different research groups, using different sample sets. And the dominant framework for European population history was that three-source model, which worked beautifully for most of the continent. Finland was treated as an outlier, an edge case, something to be addressed later when more data arrived. The data arrived in 2018. A team led by Theseus Lamnidis, working out of the Max Planck Institute for the Science of Human History in Jena, published ancient genome-wide data from 11 individuals excavated in Finland and the Russian Kola Peninsula. The samples ranged from 3,500 to just 200 years old. And they used a SNP capture approach, targeting over 1.2 million single nucleotide polymorphisms, an order of magnitude more data than the old Y-chromosome and mtDNA studies had worked with. What they found reshaped the timeline. The oldest samples, 6 individuals from an archaeological site called Bolshoi Oleni Ostrov on the Kola Peninsula, radiocarbon dated to between 1610 BCE and 1436 BCE, carried a massive Siberian genetic component. Not a faint trace, not a residual signal that required statistical gymnastics to detect. A component so large that when you plotted these ancient individuals on a principal component analysis alongside modern populations, they fell on a clear cline stretching from European populations to the Njanasan, a Siberian people living on the Taimyr Peninsula, who represent one of the most genetically isolated populations in the world. The Siberian ancestry was not Mesolithic. It was not a leftover from the earliest colonizers of northern Europe after the ice sheets retreated. It arrived at least 3,500 years ago, during the Bronze Age, carried by people whose material culture, their ceramics, their arrowhead styles, shows clear connections to groups living thousands of kilometers to the east, deep in Siberia. And then came the second surprise. If you are following this channel, you know that ancient DNA studies sometimes tell you what is not there as loudly as what is. 7 individuals from Levanluhta, a lake burial site in western Finland dated to the Iron Age, roughly 300 to 800 CE, were genetically closest not to modern Finns, but to modern Sami. The Sami, who today live primarily in the far north of Norway, Sweden, and Finland, appear to have once occupied a much larger territory, including parts of western Finland, that are today firmly Finnish-speaking. This means the genetic landscape of Finland was rewritten after the Iron Age. The people who live in western Finland today are not direct descendants of the people buried there 1,500 years ago. A population shift occurred, likely connected to the expansion of Finnish-speaking settlers from the east and south, gradually replacing or absorbing the earlier Sami-related population. If you have been watching this channel for a while, you know that ancient DNA keeps rewriting the settlement history of places we thought we understood. We are working on a follow-up that looks at 2 other European genetic outliers, the Basques and the Sardinians, to see whether the same pattern of compounding isolation holds there too. Subscribe if you want to see that when it drops. So what you are looking at, when you see those Finnish dots pulled away from the Scandinavian cluster on a PCA plot, is the cumulative result of at least 2 major processes that Scandinavia largely missed. First, a Bronze Age migration carrying substantial Siberian ancestry and likely the Uralic language. Second, a later population replacement within Finland itself during and after the Iron Age. But there is a third factor, and I think this might be the most consequential one for understanding why Finns are genetically distinct today. It is not about what entered the Finnish gene pool. It is about what happened to it afterward. The Finnish population went through a severe bottleneck. In 2014, a team at the Broad Institute and Massachusetts General Hospital, led by Elaine Lim, published a landmark comparison of 3,000 Finnish exomes against 3,000 non-Finnish European exomes. What they found was striking. Despite having less overall genetic variation than outbred European populations, Finns carried proportionally more rare deleterious mutations. Specifically, loss-of-function variants, mutations that completely knock out a gene, were significantly enriched in the Finnish low-frequency spectrum compared to Swedes and other Europeans. The numbers are stark. An average Finn carries roughly 0.16 homozygous loss-of-function variants. An average non-Finnish European carries 0.095. That is nearly twice as many complete gene knockouts per person. I want to make sure that lands properly, because it sounds abstract. What it means in practice is this. When a population shrinks dramatically, when it passes through a bottleneck where only a small number of individuals contribute to future generations, genetic drift takes over. Random chance determines which variants survive. Mutations that would normally be weeded out by natural selection because they are harmful get amplified instead. They ride through the bottleneck on the backs of the few survivors, and once the population expands again, those variants are baked in at frequencies that would be impossible in a larger, more connected population. This is not theoretical. It has a name. The Finnish Disease Heritage. Approximately 36 genetic disorders that are dramatically enriched in Finland and rare or absent elsewhere. Conditions like Mullebray nanism, Finnish-type amyloidosis, and INCL, infantile neuronal ceroid lipofuscinosis. Some of the founder mutations underlying these conditions reach allele frequencies of 1% in the Finnish population. In a global population, they would be vanishingly rare. And the bottle. The founding population was small. Then internal migrations in the 16th century, when Finnish settlers moved from the southwest into the interior and east, created regional sub-isolates. Eastern Finland and Western Finland developed distinct genetic substructure, almost like 2 populations within a population, each carrying its own enriched set of founder variants. The result is that when the world's largest genetic databases tried to categorize populations, they had to create a special category for Finns. In the GNOME AD database, maintained by the Broad Institute, Finnish is listed as its own distinct population, not grouped under European. Separate, because combining Finnish allele frequencies with those of other Europeans would distort the data, making rare variants look common or common variants look rare. I rewrote this section 3 times, by the way, because it is genuinely difficult to convey the scale of this without it sounding like Finland is some kind of genetic anomaly. It is not. It is a population whose history happened to include a combination of factors: Siberian admixture, linguistic isolation from Indo-European neighbors, a severe demographic bottleneck, and geographic remoteness. Each factor individually would produce moderate genetic divergence, but together they produced something extraordinary. Now, here is where the debate gets interesting, and where I think we should be careful about neat narratives. The timing of the Siberian admixture is reasonably well established. At least 3,500 years ago, probably a few centuries earlier, based on alder admixture linkage disequilibrium dating, which places the mixing event around 2100 BCE. But the root is still argued over. Did the Siberian ancestry arrive through direct migration across the north, along the Arctic coast, or through a longer, slower process moving through the Volga-Ural region and then northwest into the Baltic? The archaeological evidence, the spread of asbestos-mixed ceramics and specific arrowhead types, supports multiple possible pathways. And then there is the question of language replacement. The Uralic languages arrived in Finland, but when exactly did Finnish replace whatever was spoken before? Sami languages were clearly present in Finland during the Iron Age, based on the Levanluhta genetic evidence. Did Finnish speakers push north and west, replacing Sami speakers? Did the process involve substantial genetic turnover, or primarily cultural and linguistic shift? The ancient DNA is starting to answer this, but the dataset is still small. 11 ancient genomes from 3 time periods across a territory the size of Germany. We need dozens more, from more sites, across tighter time slices, before we can map the replacement process in detail. Some researchers argue the Siberian genetic component was already present in the earliest Uralic speakers and was carried westward as a package with the language. Others suggest the genetics and the language traveled separately, with the Siberian ancestry arriving through trade and intermarriage networks that predated the linguistic expansion. The difference matters because it determines whether we think of the Uralic expansion as a migration of people or a migration of ideas. There is also a timing puzzle that does not get enough attention. If the Siberian ancestry arrived around 2100 BCE and the Finno-Ugric language branch split from other Uralic languages around 4,000 to 5,000 years ago, then the genetic migration and the linguistic divergence are roughly contemporaneous. That is suggestive but not proof. Languages can spread without genes moving. Genes can move without languages changing. The fact that both happened at approximately the same time in the same geographic corridor could be coincidence, or it could be the strongest signal we have that this was a genuine population movement, not just cultural diffusion. A lot of you in the comments on our previous videos have asked about the relationship between language and genetics, whether one drives the other. This is a case where the answer seems to be both, simultaneously, but we cannot yet prove the causal link. We simply do not have enough ancient samples yet to trace the process decade by decade. I am not fully convinced by either side. The data is suggestive but not conclusive, and honestly, that uncertainty is one of the things I find most compelling about this topic. We are watching a story assemble in real time, paper by paper, genome by genome. What I am convinced of is this. The reason Finns are genetically different from Scandinavians is not one thing. It is the compounding of multiple things, each reinforcing the other. Siberian ancestry that Scandinavia never received in significant quantities. A Uralic language that created a cultural boundary, limiting intermarriage with Indo-European speaking neighbors for millennia. A demographic bottleneck that amplified the already distinct gene pool through drift and geographic isolation, the lakes and forests of interior Finland acting as a buffer against the gene flow that homogenized most of the rest of Northern Europe. Every one of those factors alone would produce a subtle difference. Together, they produced a population that stands apart on every genetic analysis ever conducted on European diversity. And here is why this matters beyond the history. Right now, today, that same genetic distinctiveness is being turned into a medical tool. The FinGen project has assembled a biobank of over 500,000 Finnish participants, linking their genomes to decades of national health records. Because the Finnish bottleneck enriched rare disease variants to detectable frequencies, researchers can find gene disease associations in Finland that would be invisible in larger, more genetically diverse populations. Discoveries about cardiovascular risk, neurological disorders, and metabolic disease are emerging from Finnish data precisely because this population took a different genetic path. The same history that made Finns outliers on a PCA plot is making them pioneers in precision medicine. The Siberian ancestry, the bottleneck, the centuries of isolation, all of it compounded and preserved in 5.5 million people living between the Baltic Sea and the Arctic Circle, is now one of the most valuable genetic data sets on the planet. 3,500 years ago, someone carrying Siberian DNA crossed into what is now Northern Europe. They had no idea what they were starting. Neither did the small founding populations who settled the Finnish interior centuries later, passing their condensed genomes to generation after generation. But the signal they left behind, drifting away from the Scandinavian cluster on a plot that did not exist until this century, is now reshaping how we understand human disease. The past is never just the past. Sometimes it is a medical record that took 3 millennia to read.